Good morning children. Welcome back to online class today. Children, I hope you all learnt well and written the exam well. In the previous class, we all learned about means of transportation in plants, isn't it? Now, we are going to learn about plant hormones. Okay? You must know what are plant hormones. Plant hormones are chemical substances produced by plants. The word hormone is derived from a Greek word hormone. The word hormone is derived from a Greek word hormone that means to excite. So this plant hormones are also called phytohormones. Okay, ma'am? Plant hormones are also called phytohormones. So, phytohormones are chemical substances or organic molecules produced by plants. What are phytohormones? Phytohormones are chemical substances produced by plants. The control and coordination in plants are performed by these substances. Okay? The control and coordination in plants are performed by chemical substances. These substances are phytohormones. In several cells of plants are capable of producing these phytohormones. These phytohormones are transported to each and every parts of the plant body to perform various physiological activities. In vertebrate animals, these several glands are present. These glands secrete hormones. That hormones produce or secrete or secreted by certain glands which is present in vertebrates perform certain physiological functions like digestion, metabolism, reproduction and so on. Now listen, what are plant hormones or phytohormones? These are organic molecules produced by plants which perform various physiological, functional and biochemical responses. Next, about the types of hormones. Types of plant hormones or types of phytohormones. This plant hormones or phytohormones are broadly categorized into 5 classes. First one is auxin. Second one is cytokinin. Third one is gibberellin. Fourth one is abscisic acid. In short, you can say what ABA and ethylene. These are the phytohormones. So types of hormones or types of phytohormones are broad. These phytohormones are broadly categorized into 5 classes, namely auxins, cytokinin, gibberellin, abscisic acid, ethylene. Okay, these 3 hormones, first 3 hormones which induces plant growth, whereas these remaining 2 inhibits plant growth. Okay, so these are growth inhibitors, these are growth promoters. Auxin, cytokinin and gibberellin, these are growth promoters. So phytohormones are broadly categorized into 5 types. What are they? Auxin, cytokinin, gibberellin, abscisic acid and ethylene. For, among all these hormones, auxin, cytokinin and gibberellin, these promote plant growth, while the remaining 2 inhibit plant growth. These two are growth inhibitors, these 3 are growth promoters. First one is about auxin. The term auxin means to grow. Okay, this is the first plant hormone discovered. Which is the first plant hormone discovered? Auxin. The term auxin was introduced by Koch and Kagan-Smith. Who were introduced the term auxin? Koch and Kagan-Smith. These Kogan, the term auxin was introduced by Koch and Kagan Smith. Okay, auxins are produced at the shoot tip as well as the root tip. Tip of the root as well as the tip of the shoot. From the tip of the root and shoot, they migrate to the other parts of the plant body. Auxins produced at the tip of shoot and root, from which they migrate to the other part of the plant body. Charles Darwin first observed the unilateral growth and curvature of canary grass. He observed the unilateral growth and curvature of canary grass. It's a type of grass in the grass coleoptile. What do you mean by coleoptile? A sheath protecting the young shoot. What do you mean by coleoptile? Coleoptile, a sheath protecting the young shoot. So Charles Darwin observed the unilateral unilateral growth and small curvature in the canary grass. It's a type of grass. Canary grass, coleoptile. Okay, coleoptile is a sheath protecting the young shoot. Now then he came to the conclusion that something is produced by the shoot tip as well as root tip, and it is migrated to the basal region, and that influence was later named it as auxin by Bent. Okay, here it is. Bent, a scientist. Listen, so Charles Darwin observed a unilateral growth and curvature in the canary grass, and he concluded that some influences there in the coleoptile. What is coleoptile? A thin sheath protecting the young shoot. Something is produced, some influence is produced in the coleoptile, and it is transmitted to the basal region, and that influence was named as auxin by Wendt. Next about Wendt's experiment. Fritz Walmart Wendt, he was a Dutch biologist. He demonstrated the existence and influence of auxin in the Avena coleoptile. Okay, he selected Avena plant as a wood plant for this experiment. In his first experiment, what he did is, he selected the Avena plant. Avena coleoptile. In this first experiment, he did lot of experiments. In this first experiment, he removed the tip of the coleoptile. He removed the tip of the Avena coleoptile. It is oats. Okay, Avena sativa. It's oat plants. It's a type of grain, isn't it? Now, in his first experiment, he removed the tip of the coleoptile, but he did not observe any growth. He concluded that something responsible for growth is present at the tip of the coleoptile. Then in the second experiment, what he did is he placed an agar block over the cut end of the coleoptile tip. In the second experiment, he placed agar block on the cut end. He did not show any. He did not observe any growth. In this third experiment, what he did is he placed the detached coleoptile tip on the agar block, and after 1 hour he removed this detached tip, and he placed the agar block over the cut end. Now it shows, after few hours, it shows growth. From this he concluded that some influence is transmitted from the detached coleoptile tip and it transmitted to the cut end and influenced the growth. From his experiment, Wendt concluded that a chemical diffusing from the tip of the coleoptile into the cut end and it is responsible for growth. He named it as auxin. Auxin means to grow. Okay. So from here, what he did for the first experiment is he did lot of experiments using Avena coleoptile or woods plant. Now, in his first experiment, he removed the tip of the coleoptile. In the second experiment, but in the first experiment, he did not observe any growth. In the second experiment, he placed the agar block over the cut end. Now, also he did not observe any growth. In his third experiment, he placed the he placed the detached coleoptile tip on the agar block. After 1 hour, he kept removed this piece of coleoptile tip and placed the agar block over the cut end of the coleoptile. So this plant shows growth. So from this experiment, he concluded that a chemical diffusing from the tip of the coleoptile into the cut end of the plant and it shows the growth. So from the tip of the coleoptile some chemical that is transmitted from the agar block to the cut end of the coleoptile and is responsible for growth, and he named it as auxin. That means to grow. Next is about types of auxins. There are 2 types of auxins, namely natural auxins and synthetic auxins. What are the types of auxins? Natural and synthetic. There are 2 types of auxins, natural and synthetic auxins. Auxins produced by plants are called natural auxins. What are natural auxins? Auxins produced by plants are called natural auxin. Example, IAA, indole 3-acetic acid. What is IAA? IAA is a form of natural auxin produced by plants. Okay. What are natural auxin? Auxins produced by plants are called natural auxins. Example, IAA. What is IAA? Indole 3-acetic acid. It's a form of natural auxin. Next is about synthetic auxin. What are called synthetic auxins? Artificially synthesized auxins that have properties like auxins. They are called synthetic auxins. Natural auxins are produced by plants, but synthetic auxins are synthetic auxins are artificially synthesized auxins, but they have the properties like auxin. These are called synthetic auxin. Example, 2,4-D. 2,4-D. That is 2,4-dichlorophenoxy acetic acid. So what is 2,4-D? 2,4-D is a synthetic auxin synthesized or this artificially synthesized auxin, that is synthetic auxin. Types of auxin, natural and synthetic auxin. Auxins are broadly categorized or classified into 2 types. What are they? Natural auxin and synthetic auxin. Natural auxins are produced naturally by plants. Example, IAA, that is indole 3-acetic acid. Then synthetic auxin, artificially synthesized auxin is called synthetic auxin. Example, 2,4-D, 2,4-dichlorophenoxy acetic acid. these you all already know that these hormones are responsible for the physiological and or functional aspects in plants, isn't it? Now we can learn about the physiological effects of auxin. Auxin brings a variety of physiological effects in different parts of the plant body. First one is it helps in elongation of stem and coleoptile. Okay, it helps in elongation of stem. Then it is responsible for the elongation of organs, or it helps them to grow. First one is elongation of stem. Second one is auxin induce root formation at low concentration. When it is in high concentration, it inhibits the root formation. So first, physiological effects of auxin. The first point is it helps in the elongation of stem, which makes them to grow. So suppose if the stem elongates, the growth occurs, isn't it? So first, physiological effects of auxin is helps in the elongation of stem. Second one is the auxin induce root formation when it is there in low concentration. When it is present at low concentration, auxin induces or promote root formation. When it is in higher concentration or when it is present in higher concentration in plants, it inhibits the root formation. So second point is root formation. Third physiological effect is or physiological function is the auxin produced by apical bud. What is called apical bud? Bud produced at the apical region or tip region, isn't it? So this bud produced at the apical region is called, suppose a plant, the bud produced at the apical region is apical bud. At the lateral sides of the stem is called lateral bud. Auxin produced by apical bud suppress or stops the growth of lateral bud. This is called apical dominance. So auxin produced at the apical bud or auxin produced by the apical bud suppress the growth of lateral bud. This is called apical dominance. So apical dominance in the sense apical bud is dominating there. So auxin produced by the apical bud suppress the growth of lateral bud. That process is called apical dominance. What is called apical dominance? So the auxin produced by the apical bud stops or arrest or suppress the growth of lateral bud. That is called apical dominance. Next is seedless fruits are formed without fertilization. So seedless fruits without fertilization is induced by auxin. When oxygen is applied externally or external application of auxin induces seedless fruit formation. So seedless fruit formation process is called parthenogenesis. What is called parthenogenesis? Partheno, or we can say parthenocarpy. That process is parthenogenesis. And seedless fruits without fertilization is induced by the external application of auxin. That is called parthenocarpy. So seedless fruits are formed by the application of auxin. That is called parthenocarpic fruits. The process is parthenocarpy. Now, this auxin prevents the formation of abscission layer. Auxin prevent abscission. Abscission means this detachment of plant parts such as leaf, fruit, branches, etc. That is abscission. Auxin prevent abscission. These are the physiological effects of auxin. So first physiological effect is auxin this helps in elongation of stem. Then auxin induce root formation at low concentration. When it is there in high concentration, it inhibits root formation. Next, auxin produced by the apical bud suppress the growth of lateral bud. That is called apical dominance. Next, seedless fruits are formed without fertilization or induced by the external application of auxin. Next is auxin prevent the formation of abscission layer, or auxin prevents abscission. What do you mean by abscission? Leaf fall, fruit fall, or the detachment of plant parts, that is abscission. Leaf fall, fruit fall, or the detachment of plant parts. Next, what is the next one? Cytokinin. So, which is the first plant hormone discovered? Auxin. Second, cytokinin. So, the word cyto means cell, kinin or kinesis division. That means cell division. The word cyto means cell, kinin or kinesis means cell division. Now this hormone, from the name itself we come to know this hormone promotes cell division. Cytokinin promotes cell division. It was first isolated from herringfish. Herringfish sperm. The cytokinin was first discovered first isolated from herringfish sperm. This next zeatin is a form of cytokinin isolated from zeamase. So what is zeatin? Is a form of cytokinin isolated from zeamase. Then cytokinin is found more or abundant in the liquid endosperm of coconut. Listen, cytokinin, the word cyto means cell, kinin means division or kinesis means cell division. So this from the name itself we come to know this hormone promotes cell division. It was first isolated from herringfish sperm. Then zeatin is a form of cytokinin obtained from or isolated from zeamase. This next is it is so abundant or it is found more in liquid endosperm of coconut. Next, the physiological effects of cytokinin. Cytokinin induces cell division, or cytokinin promotes cell division or cytokinesis in the presence of auxin. Okay, so cytokinin induces or promotes cell division in the presence of auxin. That is the first effect. Physiological effects of cytokinin. What is the first one, mom? The cytokinin induces or promotes cell division along with auxin or in the presence of auxin. Then cytokinin also causes cell enlargement. First is cell division, second one is cell enlargement. Then both auxin and cytokinin together help in the formation of new organs from the callus. Helps in the formation of new organs from the callus. You must know what is callus. Callus is a mass of undifferentiated tissue. Callus is a mass of undifferentiated tissue formed in plant tissue culture. What is callus? Callus is a mass of undifferentiated tissue formed in plant tissue culture. So the third physiological effects of or functional aspects of cytokinin is both auxin and cytokinin. This induces the formation of new organ from the callus, that is called morphogenesis. What is called morphogenesis? Formation of new organ from the callus. Formation of new organs from the callus is morphogenesis. Both auxin and cytokinin induces the formation of new organs from the callus. That is morphogenesis. What is callus? Callus is a mass of undifferentiated tissue formed in plant tissue culture. Then cytokinin promote the growth of lateral buds and it suppresses the growth of apical bud. In the presence of apical bud, cytokinin Cytokinin promote the growth of lateral bud in the presence of apical bud. Next, application of cytokinin delays the process of aging in plants. That is called Richmond-Lange effect. Richmond-Lange effect. What is called a Richmond-Lange effect? Application of cytokinin delays the process of aging in plant. Application of cytokinin delays the process of aging in plants. That is called Richmond-Lange effect. So these are the physiological effects of cytokinin. Cytokinin, what is the first effect, mom? Cytokinin induces cell division. Cytokinin induces or promote cell enlargement. Then cytokinin and auxin together induces the formation of new organs from the callus. That is called morphogenesis. Then cytokinin promote the growth of lateral bud. What about auxin? Auxin promote the growth of apical bud. Then cytokinin promote the growth of lateral bud in the presence of auxin. Then what is the last one is application of cytokinin delays the process of aging in plants. That is called Richmond-Lange effect. Okay, these are the physiological effects of cytokinin. Children, you must know this. You must learn these physiological effects of the different types of plant hormones. Okay, now turn to page 239. Children, turn to page 239. Choose the correct answer. First, question number 2. The hormone which has positive effect on apical dominance is option B, auxin. Number 3. Which one of the following hormone is naturally not found in plants? 2,4-D. That is synthetic auxin, isn't it? 2,4-D. Fourth question, Avena coleoptal test was conducted by option D, F.W. Wendt. Fill in the blanks. First one, Roman number 2, fill in the blanks. First one, dash causes cell elongation, apical dominance and prevents abscission. Auxin, right? Auxin. Then fill in the blanks. Question number 5. The hormone which has negative effect on apical dominance is cytokinin. Turn to page 240. Short answer question. Roman number 7. Short answer question. First one. What are synthetic auxin? Give examples. Answer is there in page 230. Answer is there in page 230. Artificially synthesized auxins, that paragraph you mark it. Next you write 2 more questions. Write down the physiological effects of auxin. In that page you have to write, inside the text you have to write 2 questions. One is page number 230. Write down the physiological effects of auxin and mark the 5 points. Learn the 5 points. Next write down the physiological effects of cytokinin. Mark the 5 points. Okay, children, you have to learn this short answer and fill-ups and 4 mark answer, that is physiological effects of auxin and physiological effects of cytokinin. Learn. After learning, you have to write it in the notebook. Okay, we can meet again in the next class. Thank you, children.